My first impression of NASA will come from growing up in the 80s and 90s. Astronauts and space stuff. NASA's no longer just in space. Now they're looking deep into elements that could not have been seen before. Right here on the Earth.
The measurements we make are part of a grand experiment. Just as there are sounds in nature that we cannot hear, there's light in nature that we cannot see. And our instruments are engineered to make understandable that light that we cannot see. Physics tells us what the spectrum of sunlight is, the package of wavelengths that come out of a 6,000 Kelvin star. Is a little bit that goes from 0.4 to 0.7 microns. That's the bit we can see. That's the rainbow of light. We experience that as color with our RGB cones in our eyes, and then our instruments can see all the way into the near and shortwave infrared. The amount of information is incredible. Remote sensing is something that NASA's developed to look at planets with. I recently was involved with calibrating a mission that's on the way to Europa, one of the moons of Jupiter that we think has an ocean beneath the ice. That technology that's been developed for that purpose, we've found very useful to put it into aeroplanes and take a closer look at the most interesting and the most important planet.
Biodiversity is life. It's important for us as a species because we rely on the things that biodiversity provides. The food we eat, the air we breathe, the land that we live on is quite literally maintained by the other organisms that we share this planet with.
Biodiversity loss is one of the existential crises of our time. When we lose something, a species or an ecosystem, it's gone. So it's an existential crisis, and it's also a global crisis. So global problems require global data, and one thing NASA does is look at the Earth as a planet.
We focus on everything to do with Earth. Earthquakes, volcanoes, agriculture, urban heat, vegetation, all the way up into the clouds and the aerosols in the atmosphere. My particular project is developing a biodiversity index. It's something that we don't yet have confidence that we can do globally from space. And so the way to fix that is to start on the ground.
We have the belief that we don't walk alone. We walk with the spirit of our ancestors. And when we enter these sacred spaces, we have to announce these lineages that we walk with. And we greet the spirit of the old ancestors that live in these spaces. It gives me that anchor to be able to be in touch with the spirit of the world in a way, and be able to translate that in music.
love being out here just because you can always find something new and different. The greater Cape Floristic region is off the charts from a biodiversity perspective. It's predominantly winter rainfall, and it's surrounded by sea on 3 sides and summer rainfall on the other. And so it's this isolated region that has to deal with often abundant rainfall, but then hot, dry summers with nothing. And then that's all overlain on a very nutrient-poor set of geologies. So these things are really scrabbling to make a living, and in a way that's created lots of opportunity for coming up with weird and wonderful different ways of doing it. And I think that's been a big driver behind the diversity we have here.
Just as the terrestrial environments are remarkably biodiverse in the Cape, the aquatic environments are also really fascinating to study. This is a place where 2 oceans come together. It creates this remarkably productive ecosystem, and that dynamism that happens, it's the ingredient for life. There are a lot of endemic species in the waters. These are species that are only found here and nowhere else in the world. So from a marine perspective, if you're gonna study biodiversity, you gotta come here.
If we really want to be able to monitor biodiversity from above globally, then we need to be able to do that in the most challenging parts of the world.
The program manager likes to say, this is where we'll break our instruments.
The reasons for that include very high topographic complexity, all the way down to the physical size of plants in this region is small. Fane boasts small species, small leaves, small stems, but extremely high diversity at the same time.
So this is really the boundary testing for a lot of what we need to do for global remote sensing of biodiversity.
The BioSCape projects span the full range from the top of the mountain to out to sea. We've constructed it to have a large number of field teams on the ground and in the water collecting direct observations of those ecosystems, from which organisms are there to what you can see from above.
We're working with sensors that are in development for the deployment on satellites. So the BioSCape project is kind of almost like a pilot study to see what can we do with these kinds of data.
When you look at it with your eye, we're used to seeing images in 3 channels, so a red, a green, and a blue. When we see it with a spectrometer, we see it with hundreds of wavelengths simultaneously. That transforms that from a red, green, blue image to a full waveform. So that's 400 some channels, from the visible to the shortwave infrared. And that's one sensor. We have a second sensor that takes those observations all the way down into the UV and measures them at a higher frequency that's really important for aquatic and coastal ecosystems. We have a third spectrometer that measures in the thermal range, and then a waveform LiDAR that measures structural information. We're in a process right now that's transformational for imaging spectroscopy. This is the first time these 4 sensors have been flown together, so we're going to learn a lot of new information about how they can be used.
Down departure NASA 520 is with you, passing 2,200, request heading of 10018.
Start recording.
So as we're flying, we're looking at measuring the sunlight that's reflected off of the target and back up into the instrument. Once the sunlight reflects off of our target on the ground, it's interacted with that light in some way. So there's been some absorption and reflection based on what it is we're flying over, whether it's a certain type of a tree or a harmful algal bloom. Everything has a slightly different spectral signature. It absorbs, it reflects light in a different way. So for everything we fly over, we can build this picture where we have these hundreds of channels of color that allow us to see those very slight differences of absorptions and reflections.
So you end up getting an aerial image of a site with this incredible data set. And obviously that's numbers. But what do those numbers mean for biodiversity? It's all about relating what the sensor is seeing to what we're seeing on the ground. So we have teams doing various different things to relate different measures of biodiversity to this one set of sensor measurements from the plane.
Phytoplankton are the primary producers of the aquatic environment. They take the CO2 in the atmosphere and create oxygen for us to breathe. They're the nutrition for the fisheries that we depend on. And sometimes, depending on what species are there, they create toxins that are harmful for wildlife and people.
We're looking at the pelagic ecosystem. So we've got a seabird CTD that we send down to the seafloor. When it gets to the surface, we collect water for analysis of carbon, and then we also collect the water for nutrient analysis.
The nutrients will tell us what is possibly feeding the phytoplankton, where the nutrient inputs are coming from, whether it's at the surface of the water, in the middle of the water column, or at the bottom. One of the ways we collect a phytoplankton sample is through a phytoplankton net.
This is a 20 micron net that we tow vertically and horizontally to determine the phytoplankton species that live in the water column and on the surface. And we'll use this sample to investigate under the microscope to identify all the various species.
I like the live stuff. I love photographing them. I love watching them, the way they move. That to me is always the best part about seeing what actually is in the water, what's swimming around. This is Noctiluca scintillans. Yeah, it's rolling. It's like a little monkey tail. That's what they use to propel themselves.
This project was conceived based on feedback from our local partners who said one of the most important things they need is some kind of an early warning system for harmful algal blooms which form in some of the bays and lead to massive die-outs of naturally occurring shellfish, but also aquaculture farms are affected in a big way. So our project is mapping of phytoplankton functional types. What makes it interesting is that they have the same kind of pigments and they impart a special signature that can be detected from space.
Okay, start logging, please. My primary tool is an instrument called a HyperPro, which is a hyperspectral profiler. You're looking at the properties of light and how they distinguish different things. Once you know a pure spectra of an individual species, you can use these unique shapes to definitively pinpoint individual species from space.
What we are hoping then is to develop an early warning system that can be used by local decision makers who support the aquaculture farming in South Africa.
There's so much in the coastal zone. People rely on this system for food. And then there's also the biodiversity. People are birding behind us constantly, seeing the beautiful flamingos. When you first look at this system, you see beautiful patches, but you don't discern the individual plants that are actually there, the individual species that could be so similar to one another.
Today, we are at Sandpark, west coast of Cape Town, where we have collected data for the vegetation of salt mass. The one with a tube
It's called Sporobolus maritimus. We have the Phragmites, and the one which is the shorter one is the Oxaliconia.
We go in, we measure out our plots about 10 by 10 meters, and we're going to walk transects along that, identifying every species we see on the left and right side of us, and measuring the heights of the species. We're also trying to take salinity.
You just have to take a drop and put it on that refractometer and then look inside your eye and take the reading there.
So there's a lot of indicators of these wetland environments. They can also accumulate massive amounts of carbon in them.
At the top of the core, you normally find the organic carbon. So we try to assess our core and see, okay, what type of elements we can find.
All those parameters, it has a direct impact on the signatures. The sensor that is carried in the back, this is pretty same like the sensor that is being mounted on the aeroplanes. The only difference may be the resolutions between the two sensors and also the height. So this is a handheld where you can point straight to the object, like the vegetation here in our case, and then we see the signatures on the screen. Based on this characteristic, can be used to discriminate between different species and understand the distribution and the functions of different plant species. So the device assists us to understand how the signatures look like from low level of the ground to the sensor that is being mounted on the aeroplanes.
We're trying a different approach, kind of a fast animal biodiversity monitoring approach. We collect sounds from around the region, and we're trying to get an overall sense of acoustic diversity, various noises emitted by animals, and whether the more kinds of sounds you have, does that relate to the amount of species that you have. It's a little avant-garde and exploratory. See the little checkbox there. We're really trying to do something really rapid here rather than get into a whole like species identification project. It'll be interesting to see how it all plays out.
Yay, back in the forest. This is actually the key part of it, is just this little circuitry board and a microphone. Handy-sized branch. And yeah, we should pick up a whole bunch of different things in this forest. Olive thrushes and Cape robin chats, Cape batis, more of the forest-associated things, maybe even forest buzzards.
So we see these recorders are relatively cheap, and we can put a lot of them out. And if we have automated ways to then connect that to diversity, that's a really kind of low cost, easy way to get a lot of on the ground data. And then when we link that to remote sensing data, we can broaden the scale and scale across a whole region.
In the silence, the voice of nature is so loud that it makes you humble yourself. I feel a bit sad that I didn't grow up in the rural Eastern Cape, because I didn't grow up learning the many songs that people would write, when they would go out. In the West, you would say they would compose, but really it's like they're receiving these songs from the land. They're very elemental. And even the instruments that people would play, these bows, uhati, instruments that really tied people to the frequency of the land in a way. When I came to the natural world is when I began to hear those compositions clearly. So they're a big part of my music now. Our country is so interesting because we are wealthy beyond measure. because of this natural heritage.
You cannot only look at water in isolation. It's one of the drivers of the ecosystem. So you have to look at what's happening in the environment generally, and this is why this BioSCape information is going to be very, very important.
We have an exciting tool called environmental DNA, and when I started learning about it, I was like, this is what I want to do. Let's say there's a fish scale, or there's a leaf that falls into the river. That DNA will travel downstream, and some of it gets stuck. The sediment kind of protects it, and so we collect the sediment hoping to catch a couple weeks of signal, or we catch a couple days of signal in the water that may be from many kilometers upstream. From these samples, we're trying to get an index of the entire tree of life. We usually get about 1,000 species in a single sample. Most of those are bacteria and fungi, but we also get plants, animals, protists. It's really astounding biodiversity. One of the things that we love to do is get ecological co-occurrence networks. So whenever we see this plant, we see this fungus, and we start to infer some relationships there. It's like social networks, figuring out who's the Kim Kardashian of the system. For a lot of these species, they're very rarely observed, or they're microscopic, or they're nocturnal, and because we're able to capture them and see their patterns across space and time, we might be able to learn something about them and actually give them some traits. We'll be doing some modeling of the biodiversity along the rivers. We're looking at what scales that links up with the remote sensing. So is it very local, or is it entire watershed scale?
For me as an ecologist, to be able to work here is a dream come true. The big question is, can we even characterize this diversity using remote sensing data? We can't map every species. There are dozens to hundreds of species in a hectare. And so rather than being able to document every species, we document the chemical variability. And so I'm interested in the chemical traits of the vegetation here. The easiest thing to look at is just differences in pigmentation that you might see, but there's all sorts of differences that our eyes don't see, but that the cameras from NASA do see. And so we're going to measure the chemical variability in these ecosystems to be able to see if we can use that as a proxy measurement for the species diversity. Alex, leucodendron, and Esperanthus. We go out to a plot that has been previously characterized. We know the species that are there, and we know the percentage of the species that are there. We'll collect some foliar material, put these in bags, label them, then we'll bring them back to where we're staying. We'll make spectral measurements of these, which will tell us a lot about the chemistry and the pigment concentrations. We'll scan the leaves and weigh them. We'll later dry those leaves and weigh them again. That will give us the water content. And also by having scanned them, we'll be able to get specific leaf area, which is a good indicator of photosynthetic capacity. From that, we create a metric of the chemical diversity, or what we call functional diversity. And so the imagery in the ground, it's the same information, it's just at a different scale. Here it's the scale of a single leaf or a plant. From NASA, it's the scale of a pixel. And so we'll be able to develop this concept of what the diversity is across the whole landscape.
So what we do is we pick 3 traits that we think are somewhat different from each other, and we merge them together. Red shows us nitrogen in the foliage, which is associated with higher growth rates. Blue shows us sugars and starches, which are the reserve energy sources for the plants to do their different functions, such as grow or create seeds. And then green, we have phenolics, which are defensive compounds against herbivores or pathogens. So what you're seeing here is some of the variation in different strategies that the different plant types have for allocating their resources that contributes to what we call functional diversity. It is just scratching the surface right now, and one of the things that we're doing for the science part of it is to come up with hypotheses about why and where the patterns of diversity exist. And then the other is to actually use this information to target places that might be of conservation concern. One of the really useful applications of these data is to identify locations that have been invaded by alien species.
Many of our most problematic invasives are trees. We have a lot of alien pine trees, gum trees from Australia, and wattles. They really can completely transform the landscape.
We have very high endemism, which means species that occur nowhere else in the world. And so if these aliens come in and shade them out and we lose them here, they're gone from the planet.
They grow very tall, but they also root very deeply, and so they can really reduce stream flow and reduce delivery of water to dams and for public consumption.
We need to have functioning ecosystems. You can live without love, but nobody can live without water. We cannot lose those precious services that we have now free of charge.
Economists have calculated it, and the impact runs into the billions of rands. What we found is that efforts to do restoration can often be piecemeal and unstrategic. So how do you even begin to tackle the problem if you don't know where the aliens are?
The invasive species mostly have very unique spectral signatures that we can pick up in the imaging spectroscopy. So the Abris NG instrument can really help us isolate where we're seeing pines. So from an alien mapping perspective, I think imaging spectroscopy is going to be changing the game.
So that's what our maps are trying to do, to contribute to more strategic management of invasive alien trees.
When I started work around mapping kelp, I did not really envision going this deep. I can hear my heart beating. I calm my nerves down, and then my ears kick in. It's teeming with life. It's such an extraordinary space to be in. This entire kelp journey started with a fundamental question: where are South Africa's kelp forests? You can never conserve anything if you don't know where it occurs. So I engaged in a master's program to create a comprehensive map of where kelp forests in South Africa occur. We used Sentinel-2A and 2B, which is a satellite that we could acquire the imagery for free. It comes with a lot of limitations. You can only work with a certain frequency. You can also only work with a certain resolution. But if you introduce this new technology like PRISM and these very high resolution satellites, then it expands the field of view. So I know it's going to change the game. The collaboration between Slingsby and BioSCape coming together allowed me to go out in the field and hold a spectrometer, shooting it at the different species. And then once we have a complete layer of the PRISM data, we can overlay it with the frequencies that we receive and see if we can detect different species. By just mapping them well, you don't only preserve a species, you preserve an ecosystem. So it's very important work that we need to do, and accurate data is the beginning of it. The critical step in conserving it, particularly for me, is not only through the scientific lenses. There is cultural connections to these spaces.
I feel a deep sense of an ancientness. People speak of abantu abadala. They speak of the old ancestors that we can't even name. I had previously forgotten these rituals that I had learned on the land with my father. they began to come back to me. The more time I spent at the sea, in the mountains, in forests, at rivers. And it's been an extraordinary reconnecting journey.
For me, one of the exciting things about BioSCape is just this kind of new lens on a system that I know so well. I've been studying it for decades. I intend to carry on studying it for many decades to come. But to be looking at it from a different angle like this, I find really exciting.
It's been a long slog to get these data and a lot of hard work, but it's just the beginning.
When I was a graduate student, there was Landsat, and it was available to everybody. We're coming up on a period where that will be true for spectroscopy data. There will be global spectroscopy data everywhere over the planet of the Earth over the course of the year.
I think it's a really exciting transformation in the depth at which we'll be able to observe Earth from above. Building the human capacity, the technological structure for getting ready for those data coming down the line takes years of planning, all the way to education, so that when that information is ready, there's a community of people that is ready to use it.
Education to me is a positive force in the world. It gives them this curiosity, this sense of wonder, this realization that the world really is their oyster.
The number of young people and graduate students who are going to use these data to really launch their scientific journey is what has me most excited at the moment.
There have been so many colleagues in South Africa that are so excited to work with the BioSCape data and wanting to learn about spectroscopy as much as they can. So I really do believe that the future of spectroscopy is bright in South Africa.
There are so many important questions around how ecosystems respond to environmental variability, how and when they can recover from disturbance, how they are likely to respond to future environmental change.
This is not only about creatures and things we love, but it's about us and the world in which we evolved as a species and maintaining that so that we can continue to evolve as a species.
We don't want to study the Earth just a little piece at a time. We want to study the Earth system as a whole. And now we have the tools to do that.
Having an organization like NASA coming to South Africa, it's pretty awesome. I hope that it will encourage us to see also by, hey, kuhle. Kuhle. Yeah, there's richness here.